How does degenerate binding work for MHC and immunity? Have you ever wondered how our immune system can recognize so many different pathogens? One key player in this process is the Major Histocompatibility Complex, often referred to as MHC. Today, we will look at how degenerate binding works for MHC and why it is essential for our immune response. Degenerate binding means that a single MHC molecule can bind to a variety of peptide fragments, even if those peptides have different sequences and lengths. This flexibility is vital for the immune system to identify a wide range of pathogens. MHC molecules capture peptide fragments from pathogens inside our cells and display them on the cell surface. This display allows T cells to recognize and respond to these potential threats. The MHC molecules have a unique peptide binding groove that is adaptable. This means it can accommodate different peptides while still maintaining stable binding. Think of it as a flexible slot that can hold various shapes without losing its grip. This adaptability is sometimes called molecular plasticity. Each MHC molecule prefers certain peptides but can still bind to many others. This is important because pathogens produce numerous proteins, and our immune system needs to present as many of these as possible to T cells. The more peptides that are presented, the better our immune system can monitor for infections. MHC molecules are also highly polymorphic. This means there are many different versions of MHC in the population. Each variant has slightly different preferences for binding peptides, which increases the range of peptides presented across individuals. This diversity affects three main aspects: the variety of peptides bound, the shape of the bound peptides, and how the MHC peptide complex interacts with T cell receptors. From the immune system's viewpoint, degenerate binding allows it to efficiently monitor for infections by presenting a vast array of potential pathogen-derived peptides. However, this flexibility must be balanced with mechanisms that prevent the immune system from attacking our own proteins. Some self-peptides may resemble pathogen peptides, so processes like self-tolerance help avoid autoimmunity. On a molecular level, peptide binding involves various interactions within the MHC binding groove. These include hydrogen bonds and hydrophobic contacts with specific residues of the peptide. These interactions stabilize the peptide MHC complex and influence which peptides are presented. Understanding degenerate binding is also important for vaccine design and immunotherapy. By knowing how MHC molecules select and bind peptides, researchers can predict which pathogen fragments are most likely to be recognized by T cells. This knowledge helps guide the development of targeted immune interventions. In essence, degenerate binding allows MHC molecules to present a broad range of peptides. This flexibility supports the immune system's ability to detect many different pathogens while maintaining self-tolerance.